Hi, everyone. My name is Renuka Kudva, and I'm a project coordinator at the National Genomics Infrastructure, which is a national sequencing facility based at SciLifeLab in Sweden. So, I am one coordinator from a team of several, and one of our key roles is to engage with scientists to discuss their research with them and guide them on the use of genomics as a tool. So let's do just that through the series of videos that I seek to introduce to you today. And this series is called Decode and Conserve. So, are you interested in monitoring populations of animals in a temporal context, identifying bottlenecks for breeding, studying the diets of your favorite animals in their natural habitat, identifying ecosystems that may not be obviously visible or beyond? Or are you a conservation biologist who's thinking about exploring the possibilities of applying genomics to their research? Well, you are in the right place. Genomics technologies that could make it possible and easier for you to dive into your research questions are more accessible than you might think. And as a sequencing facility, we are at the forefront of an incredible amount of innovation happening in genomics with new technologies being introduced every year and falling costs and expanding possibilities that these technologies can be applied to. But what you do as conservation biologists and your research questions are critical to protecting biodiversity around the world. And our two worlds, sadly, don't always communicate the way we should. So, full disclosure, I am a trained molecular biologist, and I am aware that some of us, myself included sometimes, can struggle to translate our expertise to questions in conservation biology, although they could be useful. And this could lead to some of you thinking that genomics may be too expensive, too technical, or too complicated, and may be better suited to well funded projects, but not to yours. So, there is definitely a gap. And Decode and Conserve will hopefully try and bridge that gap. So through this video series, I would like to seek to introduce next generation sequencing or NGS to conservation biologists, but I will not be doing this the usual way, which is starting with here's a cool technology, let's find applications. Instead, I will be starting with your research questions and thinking of tools that could help. Now, I'm of the opinion that the newest technology may not always be the best choice or the best suited for your research questions. Sometimes traditional genetic methods may be more appropriate, and sometimes doing nothing at all may be the right decision at a given point, simply because the tools may not have just been invented just yet. So, my goal is to try and help you think pragmatically when different molecular tools might make sense, given your actual research question, your resources, your timeline, budget constraints, etc. And this series will aim to cover everything from the fundamentals, what is DNA sequencing and why does it matter, where can it be applied, to different tools in genomics that might be useful for your research questions. All you need to do when you listen to these talks is have a sense of curiosity that I guess comes naturally to all of us in the scientific community. And why is NGI so invested in this? Well, because we learn with you. We would like to be more useful in our approach to serving you as a scientific community. And we hope that your journey will lead to more scientific questions and drive science forward, because that is really the need of the hour. Remember that this series isn't just about understanding the science, it is also about empowering you to make informed decisions about whether, when, and how molecular tools could support your conservation work. So, the series has been designed for conservation biologists working with threatened species, wildlife managers who make population management decisions, ecologists who are invested in studying biodiversity and gauging ecosystem health, graduate students who might be interested in exploring and applying molecular methods to their research, researchers who are wondering if genomics could help answer their research questions, and people like me as well, because when I learn more about what you do, I think about better ways in which I can serve you. And it also continues to keep me engaged and more curious about what it is that you do. So I'd like to thank you for that. So if you're ready to decode the possibilities of genomics for conservation, follow us on YouTube. And if you have any questions or about genomics in general, applying it to your research, or questions about the series, please feel free to email us at support@ngisweden.se because we are here to help and we'd be happy to hear from you. Thank you very much.